So I really want to kind of divide this talk into two different parts. The first discussing a little bit of facts and a little bit of my opinion, just some overarching concepts of how do we best implement digital data management and data management in general. Each of these topics could probably be like their own workshop or symposium or really day-long course. But I'm going to just, of course, brush over them quickly. So sorry about that. And then we'll talk about what I actually work on and how that relates back to some of those topics that I introduced at the beginning. So why do we need digital data capture management? We've heard a lot of talks already today, and it's pretty much impossible to implement a lot of those technologies without some sort of foundational data management in place. For the genomic selection stuff, for instance, if you want to utilize your previous phenotypic data, you have to know where that is. You have to know what it is actually talking about. If all of your fields start at plot 1 and go up to plot 1000, how do you differentiate plot 13 in this field from plot 13 in that field? You need to be able to track those things as you're collecting them, as you're using them, to enable that data to actually be usable later on. So, second reason is speed. It takes a long time to collect phenotypes. Kai attested to this. I don't even remember how many phenotypes they were collecting, but a ridiculous amount. Doing that all on paper or manually, or having somebody tell you what to write down, and then having to retranscribe all of that information later on, takes a very, very long time. So by integrating these tools into breeding programs, we can shorten the amount of time that it takes to collect those phenotypes, thereby really, letting us collect more, except the productivity gains are going to be offset by the bigger populations that we can have, so you're still going to spend the same amount of time in the field. So I can't say shorter field days, but more populations, bigger populations, bigger numbers of entries, which we really need to enable the gains to facilitate the increased amount of food that we need to produce for the world. And last but not least, accuracy. So as I mentioned, every single step between observing something and actually making a decision on that something is a place where error can be introduced. If we just choose a really low 1% error rate on saying, transcribing, or scribing, reading, retranscribing into a computer, and then analyzing, we have roughly 5% random data by the time we actually start doing the analysis and making our selection decisions. Cutting out each of those steps increases the amount of genetic gain we can affect, and really just lets us do a lot more. So the big concepts. How do we actually do some of this? It starts out with an enormous amount of text describing databases. So what is a database? Who here uses like an actual database, not Dropbox with Excel files, for their breeding program? Yeah. No hands.
Crickets. There were crickets.
There were crickets. You need to, and I know like it's hard to learn about something that you know nothing about. That's what we're all kind of doing here today to some extent. But so a database in and of its like the bottom essence is just a bunch of tables that relate to each other, that each store unique records. We have different columns that are attributes about each of those records, the rows being the records. And then we can kind of start joining those tables to relate the concepts to each other. So for instance, the examples I give here are we have a table, say plot or location. Within that table, we can have a row with a plot ID or a location. And then we'd have different attributes about each of those things. So for instance, this plot might have an attribute, what variety is this plot? What experimental entry is this plot? Whereas the location for Obregon would have where are the coordinates of this actual field? What treatments would go onto this field? Just all of the data that you know, somebody else doesn't. And there's a lot of times where someone else is doing the analysis, but doesn't quite get all of the data that you know before that analysis is done, and it causes a lot of problems. So I think you should view databases as, there's a lot of push to have them as this like cold storage where you just archive everything, but they're more of a vault, in my opinion. And you don't necessarily put only the things that you want to lock away forever in the vault. You put whatever you think is important in the vault. And if you're collecting phenotypes on something, if you went through the effort of planting and organizing and maintaining a field full of genotypes, each of those phenotypes should be important, and you should want to retain that information. So this is the broad strokes points. There are commercial and there are free options. So my 4 bullet points on setting up your own database. Think about what you need. Design that rigid framework. Populate it. Use it. Make sure everyone in your program is using it. So add records as you start needing them. This was one of the enormous advantages of seeing. I was one of the first students in the Poland lab, and this was an enormous advantage of being one of the first students, was seeing how this database, we just have a simple MySQL database, but how it was set up, how it was organized, the power that can come from that database. And then if you need more resources, there are online courses. YouTube is always just a great place to start. But all of these things you need to do to get the full benefit from the data that you're collecting. The biggest point that I didn't even put on the slide, of course, is you can't break anything. Like, you're setting up, you're experimenting. If you break whatever you're setting up, it's okay, because you didn't start with anything. Don't be afraid of breaking things. That's like the biggest point in, I think, research that we all kind of miss a lot of times. If you don't do it right the first time, if you do it like 40% of the way right, that's 40% more than you already had. So you just learn, you iterate, and you make progress that way. So expanding on some of the different characteristics of what can go into a database. And then what I kind of think is important for a breeding program is the introduction of unique identifiers. And so these, in my opinion, just half opinion, half fact, of course, are vital to a breeding program. Every single thing that you are collecting data on should have a unique ID that is unrepeated anywhere else. There should be no 2 plot 13s that there's any ambiguity of where that data came from. So we needed to know exactly where and when a sample originated. Now, there's a few different ways to generate something that's universally unique. What we actually use in our lab is kind of a structured unique ID. So we combine known elements of the individual just into a string that we then use to reference that individual. Say the downside to this is that it takes some mental awareness. So you need to know that you're not repeating things. For instance, if like our plot ID earlier, so from that plot ID, I can tell you what year it was planted, where it was planted, and then just an arbitrary number corresponding to that plot. The arbitrary number is where things might get tricky, because if you have 10 people who are designing plots, then you all need to coordinate to make sure that you don't overlap, because we want something unique, not overlapped with anything else. So kind of with that in mind, one of the things that I've been leaning toward is using, I said random, but it's more of a universal unique ID generation system. And this website, uuidgenerator.net, is great for that. And it just, you go there, you say I need 50, 500, 50,000 unique IDs, and it gives you an ID that is universally unique. It doesn't correspond to anything, but it's not going to be repeated anywhere else. And so this is an example of one of these IDs, which is a lot more difficult to extract meaningful information from, but it really doesn't need to be. For all practical purposes, we don't need to ever see that. We just need to know that it exists so that we can eventually trace back to that if we need to. So, along with unique IDs, the introduction of barcodes. This is the Wikipedia definition. Everyone knows what barcodes is, or barcodes are. But having the ability to tag plants, plots, pots, seed bags, germplasm, everything with a barcode really facilitates not only collecting data more quickly, but eliminating one of those steps between observation and collection. And so we can do this a couple different ways. So we can use barcodes in the way that they're more designed. We can use them to identify individuals, but we can also utilize them, which I'm going to talk about in about 15 minutes. We can also use them to collect phenotypes just by relying on their inherent nature as well. But integrating barcodes is one of those steps that will, if you're writing down every unique ID, you're going to make a mistake at some point. If you're writing down anything, you're going to make a mistake at some point. So eliminating paper and pencil is kind of my ultimate goal from breeding programs. All right, so with that in mind, kind of the optimal scenario for breeding data: minimal manual data handling, having some sort of database, having unique IDs, integrating and actually using your barcodes, using being the big qualifier there. If you have barcodes but you don't use them, I mean, what's the point? You're wasting time making them and wasting time doing whatever you're doing not using them. And then finally, having the tools to facilitate data capture. This is what I'm going to spend the rest of the time talking about, because this is what I actually worked on. And so for us, those tools are apps. So I'm going to talk about 4 apps that we've developed so far, kind of how to use them, a brief overview, summary, etc., and then kind of where they could fit into your breeding programs or to your research programs, giving some examples. Yeah. So the first app is Fieldbook. So one slide summary. This is an app we designed for, I'm going to go through this. This is like 95% of the talk. So one slide summary though. This is an app we designed for phenotypic data collection. Originally designed it in 2012, but we've been building it, expanding it, adding new features based on requests from users all over the world. But it really simplifies how data is collected. Allows you to navigate between plots or traits, but it also shows extra information while you're collecting the data. I need to reorganize this. You can manage the traits within the app. You can create custom traits depending on what you're actually collecting. It's been translated into 12 different languages by all volunteers, people who have reached out and said, I want this in Arabic. Tell me what to do. It's exceptionally flexible, and when I was putting this together, we have 6,500 downloads, which blows my mind away. I gave a talk in Tanzania a month ago, and it was at 5,900. It's ridiculous. So it is actually used. I'm not just like making things up. That this is something that you could use and should use to sell you. This is the only advertising that's done for this app is me talking to crowds like this, and then it's all been word of mouth. All right, so using Fieldbook, going to go through kind of step by step on what you need to do to bring data in, to collect data, and then get data out. So I'm not going to cover this since we're going to go through each of these anyway. So, kind of small text, but that's okay. First thing that we need to do is to create and import a file. And so we really wanted to make this compatible with as many breeding programs as we could, so they didn't have to change too much. My dad's actually a plant breeder, and so I know how non-amenable to change they are. And so this is really, really close to what your field book would look like if you were going to print off a field book, go out to the field and collect data. Wanted to make it as close as possible. So there's only one extra column, and that is in this case, our plot ID. One of the things that I know about breeders is that every single one of them does things differently. And so the column names are all up to you. There's no really structure. There's no required structure built into the app outside of the 3 columns that you need. So we need a unique identifier. So we need to be able to tell the app what data goes to which plot. And that's why every plot needs to be different, whether that's between fields, between years. We need 2 columns, which I like to call the primary order and the secondary order. In this case, it's row and plot. This could be range and plot. This could be row and column. Up to you, whatever you want. And then we have all these extra columns. So this is how we bring in that extra information that, I'd love to say as breeders, but I'm not a breeder anymore since I was like 16. But all that extra information that we want to see while we're walking through the field. And in this case, I have a pedigree, the seed name, and then the seed ID. And this is the sample file in Fieldbook, which was actually one of my fields when I was doing my PhD. So when we import that file, might be difficult to see in the back. We just choose the 3 columns that we've selected for those attributes. So our unique identifier is going to be our plot ID. Our primary order will be our row, and our secondary order, our plot in this case. So we've created our field. Now we need to create the traits that we actually want to collect after this slide. So we're going to manage our fields first. And so we have our list of fields. And then we can toggle between different fields. We can import all of the fields that we need for a given season. Each row on this is impossible to read in the back, I know. Each row is a separate field here. And then within that row, we have the date that it was imported. If we edit that field, if we collect data, it'll show that date. Once we export that field, it'll show that date as well, so we can track kind of when our fields have been updated, when the data was last exported. And at the very bottom of each of these rows, we have the number of entries for that field. Now we can create our traits. So as I said, within Fieldbook, you can manage the traits that you're wanting to collect. You can import and export existing trait lists, so you really only need to create the traits once. You can add new traits directly within the app, which I put on the other one. And yeah, there we go. So you can add new traits within the app. So depending on the type of trait that you're adding, you can modify the name of the trait, the format. Different traits have different things that you can restrict. For instance, a numeric trait is going to have a minimum, a maximum, which aren't required, but just for that added level of data integrity, you can add as well. And so when we have our trait list here, We have each row being a separate trait. We have our category, our format of that trait, numeric, which I'll go through all of these in a slide or two. And then we have the trait name over here. So we can reorder the traits. We can drag and drop the traits as we need. We can, if we only want to collect 3 traits at a given time, we can uncheck all of the traits and just check the ones that we want to collect. And then we can sort them by the name, the visibility, and the format. And if we want to collect the same trait at, say, multiple time points, we can copy that specific trait to where it retains all of the qualities of the parent trait as well. Yes?
So does this app has the autosave option in real time as well? So for example, if you record the data for one plot, will it save it automatically, or you have to save like after taking the notes on 30, 50 plots?
So there is no save button in the app. It is constantly writing to the database. So whenever you collect a data point, that's the save button. So it's, yeah, I guess I could call it autosave, but it's just kind of the nature of the app itself is that it's extremely difficult to lose data.
Yeah, I was actually so scared last year. We just start first time using the KD Smart app for our breeding program, and I didn't know that whether it's gonna save automatically or not, and I was like so scared. Might be I was just losing some of my data, and by the end of the day there will be nothing.
Yeah.
But I found that I probably saved the data automatically.
Okay. So once we have our fields and our traits, we can start collecting data. So this is the main screen of Fieldbook, and it's really can be broken down into 4 different parts. Up at the top, I call these info bars. Great name. It's because they are bars that show information. So all of those extra columns that we brought in, our pedigree, our seed ID, our plant name, we can change to any of those up here. So we can see our pedigree while we're collecting data. If it goes off, if it scrolls off, you can press it and it'll expand to the full pedigree or full text of whatever thing that you're wanting to see. I use examples, but like I said, it's very broad in terms of what you can put.
Okay, question. You move forward the next plot, you just press arrow, or will it be the enter button to just use to move forward?
We'll get there in like 2 slides. Second part of the app is the trait section. So we have our current trait that's being collected, in this case height. If we want to see all of the traits, we can click on height, we'll get a trait list. Below that, we also have trait details. So in this case for height, it's being collected in centimeters, and so that's displayed there. If we want to go forward or backward to the next or previous traits, use the arrows. Very simple. Below that, we have our entry navigation. And so the forward and the back arrow move between different plots or plants or whatever you're collecting data on. But in between those is where our primary and our secondary order come into play. And so these are 2 columns that are always going to be visible to the user when they're collecting data. So I will always know which row and which plot I am on, which is what personally I use for navigation when I'm collecting data out in the field. And then the last and kind of the most important aspect of Fieldbook is the entry navigation section. And we wanted to make this really big because I lean over my phone all the time, and I don't want to do that while I'm out in the field collecting data. So half the screen is dedicated to just data entry. At the bottom here we have an NA, so we can differentiate between missing and null data, and we can delete any data that we collect as well. And so I know that not every trait that you're collecting is going to be numeric, and we knew that when we started. So one of the things that we built into the app is different formats for different types of data. And so we have numeric over here with just a simple number pad, categorical, where we can define up to 12 different categories that we want to collect and then select a single category. We have percent, where we just display a percent slider. Date, which is really useful for things like flowering, where by default the current day is shown. We can go forward or backward in single day increments. So if you didn't go out to the field yesterday, you can still collect that data. Boolean, just a true or false button. We have photo, where we can collect multiple photos of the thing, plot, that we're looking at. Those photos are named with that unique ID of the plot, so we know what the photo is of. The name also includes the name of the trait, so if we're looking at something specific, like a rust score, we will know that that photo was supposed to be of that plot, of a rust, at this time specifically. And then we can feed that into either image analysis, other tools that are not going to be talked about today, or use it for whatever we need to use it for. On the second row, we have a text trait, which just shows a text keyboard, simple kind of notes approach to taking data. We have a disease rating trait, which is also target audience for once. No one is ever the target audience for this trait. So we have a customizable 21 button panel where we can define the severity. Yeah, the severity of the disease. You can also, so you can customize that. I know people do 0, 1, 5, and then increment by 10. Some people do increments of 5. Once again, every breeder does things a little bit differently. But then we can combine this R, M, and S, which, once again, super small in the back, apologies, for the reaction type as well, so that we can simultaneously collect the severity and reaction type. If we have a heterogeneous response, we have a slash to where we can add those as well. Otherwise, you restrict it to a single percentage. Yeah. So third is a location, just a point location of whatever you're looking at. It's actually asked for by ecologists. And then moving on, we have a counter with a big +1, a small -1. So if you're out in the field counting things, this replaces the little clicker that everyone, I've never actually seen one, but I know people use them. Okay, we have a multi-categorical trait where we can define as many categories as we want, and we can combine those categories. Basically just creates a keyboard. These are, in this case, we have yellow and purple, axle and terminal, long and short, round and wrinkled, and yellow and green, which are all the traits that Mendel was collecting. And so if he had had this, it would have gone so much faster than writing every single thing down. And then last but not least, we have, actually kind of least two, we have an audio trait that records your voice as an MP3 file, which you can go through later on if you really want to. I've never found someone that, we built it in, and I just don't know who uses it, but people always, you can't take things away in software development. So once we've collected data, we can export that data. And so once again, to make this as flexible for as many breeding programs as possible, data is exported straight to a CSV file, which you can move to your computer, do analysis on. I'm going to go over the two formats that we have for data export on the next slide. But we have different options for what gets it exported. So when we imported that field file, we had all of those extra columns. So we can either choose to only export our unique identifier column or bring along everything that was originally imported. Similarly with traits, we can only export the active traits or bring along all of the traits that we have. By default, the field name includes the date and the original name of the imported file. And there's also an option to overwrite a previously exported file, which doesn't actually overwrite, it just moves it to an archive folder. Never delete anything. That's a life motto. But all of this is, you can change the file name and everything else. So for our export formats, we have two, and they differ just a little bit. We have a database export, and in this case, every single row is an observation that was collected. Now what this lets us do is also display and collect some metadata of what was being collected about the data. Yeah, that works. So in this case, for this plot 13 RPN 1, we were collecting height. We were collecting, the value is 75. I collected it in 2014 at this specific time point. But every single observation is going to have those pieces of metadata associated with it as well. Now, our table format is something more traditional, what you would probably use for your analysis if you're feeding this into, say, R or SAS or any kind of analysis software, where every single row is now a plot, just like our input file originally started out as. But we have these added columns with the traits and the values that go along with those. So over.
A really good question.
Yep.
With your metadata, do you get GPS coordinates of the plot?
Not currently. So the biggest limitation of getting GPS coordinates of each of these data points is it's kind of battery life more than anything else. Would be number one, and then super close behind that would be the inaccuracy or imprecision of the GPSs built into cell phones and tablets.
Since you brought up battery life, that's one thing that I wonder about, because I'll be in the field, say, 12 hours. There's no way a tablet's going to last that long.
You eat lunch?
A lot of times out in the field,
Yeah.
because I'm at a remote location.
Yeah, and that's
So-
common. So these were originally developed for developing countries in actually Eastern Africa. And what a lot of people have resorted to doing is charging during lunch, either with a battery pack, or if you have a vehicle there, just with that as well. But the devices that are available will last 8 to 10 hours. I would be comfortable
When
making
they're
that
new.
claim. Pardon?
When the devices are new, not
Yeah.
so much when they're old,
Yeah,
or a year or two old.
yeah.
That can be an issue.
And then one question right in front of you.
Yeah, is there an option for the barcoding data? For example, we are using barcoding for plant height measurement.
Yeah,
So there
2
is
slides.
option for that?
Everyone's just like 2 slides too fast. So as this project has evolved, we've gotten lots of requests for additional things to be added, and I've kind of lumped all of those together on this slide to some extent, talking about these 3 on the next slide. But we have a tutorial built into the app that will guide the users through how to use everything on the first startup when you're first running it. We have the ability to go to the next entry that's missing data. Once again, really good for flowering notes. Since you go out at multiple time points, the fastest way to find the next plot that you didn't have data on already is to hit this button, and now you're on that plot. You can walk to it and see whether or not it's flowered, and just rapidly move through the field instead of having to identify every single entry. Our primary order comes into play, finally, with our primary order sound option. I know, at least in Kansas, in our fields and at Summit, this was a big requested feature because fields are planted with no space in between. So sometimes you will be collecting data moving along a row, and then 5 plots later you realize that you are now in a different trial because you didn't remember to go forward 1 row. And so with this, when your row changes to number 2, it beeps at you. So you have some sort of auditory response to move forward to the next row. And then this is kind of a really useful tool for people who are wanting to collect more than one phenotype at a time. Cycling traits advances entry. So if we are collecting 3 traits on every single plot, instead of having to move between our plot and our trait arrows, we can move only through our traits And as we cycle through that complete list of traits, we will move to the next entry as well. So when my boss, Jesse, was collecting some data for one of the studies that he was working on, he had a device out in the field and said, it keeps moving to the next plot before I put data in. So this is Jesse's setting right here. If you want to have a data point on every single plot that you are collecting data on, you can enable these options, and then you cannot move unless you have a data point on that plot. And then these last three I will talk about here. So we have a quick go-to. Changes these two numbers, the primary and the secondary order, on the main screen to an editable box. So now if we want to, instead of either pressing and holding or tapping to row 25, we can just type in 25, takes us right to that row. Or if we know the plot number, do the same thing as well. We have our move to unique ID. The names are great, I know. We have two different options with this approach. This is the text option, but we also have one that takes you directly to a barcode scanner that uses the device camera. But what this does is that if we have all of our entities barcoded, we can scan that barcode, and it moves us to that unique identifier. Even if you only have like 10% of your plots barcoded, you can walk to a specific part of the field, scan that barcode, and know that that is exactly where you need to be to collect data on that plot, and then just move to whichever plot you want to go to next. And this is still kind of in development, but this is the data grid option, where we bring in that whole thing that we're trying to eliminate with the spreadsheets and making everything complicated, but lets us visualize what data has been collected for what traits on which plots. So we can scroll up and down, select a plot, and move directly to it as well to kind of just quickly see what needs to be collected and where. Okay, so talking about barcodes. So USB barcode scanners with the apps that we have, and kind of Android in general, will plug directly into the devices and serve as keyboards. So we can utilize that functionality for a couple different things. The first being height sticks. So we have barcodes at centimeter intervals, intervals. I combined them both. And then we can put that stick in a plot, scan the barcode of the height of that stick or of the plants, and then not only does it collect that height, but it'll move us to our next plot if the barcode scanner has been configured to do so. The other thing that we've used these for is a millimeter ruler stick, ruler thing. You can't really have a millimeter ruler. Laying the leaf across these barcodes, which blocks out the barcodes. So we just start from the bottom, go up until it scans, and now we have the length of our leaf. Okay, so that covers most of Fieldbook, and just talking about how to integrate Fieldbook into, I think it's kind of clear how to integrate Fieldbook into a breeding program, but just to kind of give a summary of Fieldbook. So as we observe phenotypes, we can digitize those directly. We can collect that data automatically. We can do this in the field, the greenhouse, and there are people who are doing this for phenotypes in the lab as well, just because they found that it's easier to enter in data using this than anything else that they have. There are people that are unarchiving text data into Fieldbook versus using other approaches just because it's that easy. And just as important, in my opinion, is the fact that we can organize the media that's being collected from these plots. So specifically thinking about photos, since that is just an up-and-coming area that's going to be extremely important in the future. Okay, so not all of our data that we're collecting is going to be in the field. We also have things in the lab or the office, maybe not so much in the office, but in the lab at least, that we need to organize and weigh. So inventory is an app that's kind of more, so I like to think of Fieldbook as an organized data collection app, but inventory is more of an ad hoc collection app where we don't have that structure built into the system. So bags of seed, when we need to weigh all of those or when we need to organize those into boxes, being able to do that is kind of what inventory is the best at. And so not only can we integrate our barcodes that we talked about to scan these simultaneously collecting the weight, but we can group those by different categories, which within the app are called boxes. And then again, we export this as a CSV flat file, just so that it can go wherever it needs to go to make your life easier. So this is our friend Bikram Gill of K-State, how to implement this into, or different ways to implement this into a breeding program. So organization, so within a gene bank or within any kind of inventory system, you're going to have some sort of grouping characteristic, whether that's a box or a bag or a room. Out of examples, but we can organize those. We can organize those individuals based on that, just so that we can track where they are and use that tracking later on when we need to go back and find those individual samples. The other thing that we can do with inventory is the collection side of things. So this is from David Bonnet when he was still at Summit. They had these enormous bags of samples that they needed to weigh, and implementing the inventory into the program changed the data turnaround time from a month to 2 months to just a few weeks, just because they were able to go through all of the samples so quickly. So without having to organize anything, we can collect the barcode and the weights and then have that data ready to go for analysis. Okay, our next app is called Coordinate. And this is an app broadly to organize samples into grids. It allows the user, this is like the one that I struggle the most to convey the importance, but like the coolest one too, in my opinion. So we can create custom templates where we can add different parameters to, say, exclude rows or columns depending on what data we're wanting to collect. We can change the naming schemes, so our columns and rows can be either alphabetical or numerically named. And then we can add custom data fields. So if you're collecting, so you can collect all the metadata that you're interested in and bring that in to, or have that available when you export these grids. So we have two default templates built in. We have our DNA plates, which is this 8x12 96 well plate that we use, that everyone uses, to collect tissue. We, once again, have our barcoded, theoretically, we have our barcoded samples. I don't have a picture of it right here, but I did earlier. So that we can scan the plant's barcode, collect that tissue, put it into this graphically highlighted well, and then be able to move on to the next one. So once again, kind of an ad hoc data collection where we just do things as they're being collected. So then implementing this into a breeding program is primarily going to be used for tracking the tissue sample collection into these wells. But really the flexibility of this app is kind of why it's my favorite, because we can really use it for any kind of grid that we're putting things into. And so here, this is a automated planting hedgerow planter that we use in Kansas at least. And as we fill those wells, we can track which samples go where so that we can then know what's actually in our field. Okay, so the next app is one of the newest to the collection. It's called Verify. And this app is primarily used for data viewing and verification. The workflow is importing a list of entries and data, selecting which column corresponds to your barcode, because barcodes are important. I have to reiterate that throughout the talk, how important barcodes are. And then scanning the entry barcodes to then either modify that list that you imported or to look up information about a specific entry. And so Verify has 5 different modes that are available. So by default, it'll just show the entry info. This was originally designed to import a list of GeneBake entries and then be able to walk up to an individual, scan it, get all of the information for that individual, and just have it quickly accessible that way. But it's evolved based on just what was available to have these additional modes as well. So we can match the entry order. So if we scan something out of order, it beeps at us. Really annoying beep. We can filter to remove scanned items. So if we scan something, it's removed from the list. And then we can kind of part this list down to see what's left over at the very end. We can do the same thing with just painting the scanned items to where anything that's scanned is highlighted, and we can slide or scroll up and down to see what has or has not been scanned. And then finally, there's also a pairing mode where you can assign a pair column to connect 2 individuals together. So if you are, for instance, splitting up a bulk sample into, well, that's on the next slide. So yeah, I'll talk about that on the next slide. So for these 2 modes, we can also export that modified list of entries so that we know either which samples are remaining or which ones have been successfully scanned. Okay, so utilizing Verify, Bikram's back. We can display our GeneBake sample information. So we can load all of these entries into the app, have all of the data for each one of them ready to go. Once we scan a single entry, we'll have every single data point we would want, characteristic that you pulled from your database that you set up 20 minutes ago, 30 minutes ago. So we can match order. So if we're organizing a list of samples to plant, like this, very small trial right here, we can scan through each of those entries, and if anything is not in order, it'll beep at us. Something that we originally tried with a different app, but this app just worked a little better. We can use the filtering and coloring for sample collection or for planting as well, where we have a list of samples or a group of seeds, scan the barcode of each one as it's planted, making sure that every single thing in our list is present and is planted at the end of it. And then we can use the pairing for dividing bulk samples. So if we, say, have a tub of seed and we want to split that up into specific jars or envelopes or anything, we can make sure that we scan the correct tub, and then that's followed immediately by scanning the correct subdividing container. So the ultimate concept for this project is to kind of take this consumer hardware, pair it with some inexpensive additional peripheral hardware to be able to collect an enormous amount of data and do so rapidly. At these kind of things, though, I always like to talk about what we're working on in the future, too. And so that kind of revolves around a bunch of other hardware that I'm going to start with talking about 1KK. So this is an app that's been in development for a while now. Image analysis is hard. That's the long and short of it. But the name comes from the 1,000 kernel weight that everyone's familiar with. So this is a seed image analysis app. The idea being that you lay the seeds on a mat, take a photo, the seeds are counted, it's hooked up to the scale similarly to inventory, and then we can get the average per seed weight. use that to kind of calculate a pseudo 1,000 kernel weight as well. But simultaneously, we're getting all of the length, width, morphological parameters about each of the seeds. With the possibility of exporting the sample summaries and then exporting each individual raw seed as well. So eventually having the ability to add some sort of selection mode where you set the threshold of what you want for either 1,000 kernel weight or a seed size, and then quickly being able to divide whether you want to keep the sample or throw it away. In progress. We have a master's student who this is his only project right now, computer science student. And he's making a lot of progress. We changed the workflow a little bit, but I'm not going to go into that right now. But it's looking really good. So this is another project where we just need to bring all the pieces together. But this is an app called Survey, and it's going to interact with Fieldbook. And this kind of goes back to the GPS question that somebody asked. That, yeah. So, yeah, you. So the phones and tablets GPS isn't accurate enough to really get plot level data. The precision is at the highest in the like 20 to 30 feet, but it's all over the place. If you've ever tracked your run with a phone, then you see that it just jitters a lot. But with these external, now RTK GPS units, we can utilize those not only with separate apps, but then within Fieldbook as well. And so the concept here is that we'll walk through this field using this RTK GPS to get an individual point for each of these plots. We'll have our orange commander-in-chief collecting data throughout the field. But then we can utilize the heading of the GPS along with these points to then have a pseudo impact zone of where that person is looking while they're collecting data. Now, if they look a different direction, then that plot is what will be displayed on Fieldbook. So we're really taking out that manual navigation within Fieldbook and just automating everything, because even manual navigation is one of those places where you can introduce error.
Do you have to have a base station for that?
For that one, yeah.
Okay.
Yeah, you buy, I don't remember the cost off the top of my head. I think it's like $1,500 to $2,000, but it's about 5 times less expensive than it was when we started coming up with this idea. So it's becoming more feasible to introduce this into programs not in Kansas. All right, so finally the last thing that I want to talk about that's really been, I mean, this is like really early, but we're hoping to make a lot of progress this year as well, is utilizing this crowd-funded portable near-infrared spectroscopy device to enable a selection tool for breeders. And this is all about cassava, but having the structure to have this to implement these prediction models into will really let us kind of, just having the structure there will let us add more prediction models to where we can bring this to more crops as well. So this is work being done by Jenna Hirschberger at Cornell. And the big thing is replacing expensive and laborious phenotyping with something that has high predictability. Still in the validating the model step for this, but we're still going to move forward with developing this app because it has a lot of potential. So for our project specifically, one of the big things, one of the big components is just the outreach and the training. I've been at a bunch of workshops over the last 6 months, or hosted, organized. And part of that is just to make people aware of these things, but also to bring a better understanding of what we think is good ideas to people. But So the project website is phenoapps.org, and on the website we have information about all of the apps and the hardware. But we also have this WordPress Coursera somewhat thing built in to the website as well. So we have courses that are completely free, kind of walk you through step by step, similar to what I did with Fieldbook, but a little more in depth as well, on how to use these different apps. And then that's kind of keeping those up to date with the current updates of the apps as well, so that any changes you can go back and reference these courses. Okay, project website. All of the apps I talked about, the first 4 apps I talked about, are available on Google Play. Everything's free, open source, on GitHub, and then this is our Poland Lab webpage as well. And I think all of these presentations are going to be available, so. Acknowledgements, I'm in Jesse's lab. Mike, Jenna, and Paula are working on the cassava side. We have a collaborator in Uganda who's also doing some image analysis with the cassava. Enghua and Dory have provided funding for different aspects of things along the way as well. And with that, I would be happy to take any questions.